In this video, we're going to finish the topics from chapter 3. Understand that the ability of amino acids, peptides, and proteins to have different charges at a given pH is very useful. The important application of the isoelectric point relies on the separation techniques that can be utilized in order to obtain pure proteins for experiments. Understand that these techniques are not exclusive. They have to be used in combination with other techniques in order to obtain the pure protein, but it is involved in the analysis of samples, for example, in biochemical reactions. Understand that electrophoresis is a common method for separating molecules in an electric field. One of the things that will be discussed in later chapters, specifically chapter 5, is how the isoelectric point for a particular protein can be utilized in techniques in order to separate samples. Just to give you a brief introduction, here we have an isoelectric focusing gel. In this isoelectric focusing gel, what we can see in the image that we have on the left side of the slide is that we are going to have a sample of proteins that is loaded onto a gel, and because we have different pHs present in that gel, then depending on what is the isoelectric point for the sample that you have, it could be amino acids by themselves, it could be peptides, or it could be proteins, they are going to be separated. Once that separation occurs on pH, then it can be used in combination with other techniques, in this case electrophoresis, in order to look specifically at the protein of interest. So this is just an application for the isoelectric point. Now we're going to proceed to discuss the topic in section 3.4, which has to do with the peptide bond. So far, we have discussed the properties of amino acids and how do they behave individually. Understand that an individual amino acid can be linked by forming covalent bonds. This covalent bond that is going to link two amino acids, it's going to happen specifically at a particular area of the amino acid. So if we focus specifically on the top figure on the left side of the slide, we see that we have amino acid 1, which I am using the abbreviation AA1, and we have amino acid 2, that's AA2. So when we're trying to combine two amino acids, understand that the reaction that occurs, which in essence is a dehydration reaction, it is called amidation reaction. And in this amidation reaction is how a peptide bond is formed. If we look at the functional group that is present in a peptide bond, we see that this is an amide bond. If we think about in organic chemistry, the way that amide bonds are generated, there's a wide array of reactions. And in those reactions that were discussed in previous classes, you understand that having a carboxylic acid and an amine may not be the most advantageous way to produce this amide bond. But understand that in this class, we're talking about biochemistry. The formation of this peptide bond or this amidation reaction happens because of biomolecules. Under biological systems, as we're going to learn throughout the semester, the rules of organic chemistry apply in terms of the functional groups that we're making, but because of the molecules or the biomolecules that are utilized for the reaction, those functional groups that may be difficult to make in the laboratory, nature has a very efficient way to make them. So understand that when we're linking two amino acids, overall in the reaction, there is a loss of water, but we're going to be forming an amide bond. So That is the functional group that is present in a peptide bond. It is important to note that the peptide bond actually has a resonance structure, and we can see it in the bottom left corner of the slide. Okay, so understand that in that peptide bond, which I'm going to highlight, in our resonance structure, because the nitrogen atom has a lone pair, those electrons can actually move, generate a carbon nitrogen double bond. Then this is going to make the electrons go up to oxygen, and that's how that second resonance structure results from. So one of the things that is important to note is that then this nitrogen, it is actually quite planar, because if we look overall at what is the electron group geometry for that nitrogen, this is going to be trigonal planar. So overall, it is understood that there is no rotation about that bond that is formed due to resonance. Understand that when amino acids are linking to one another, that results in the formation of a peptide. And these peptides are going to be formed by linking a small number of amino acids. Okay, so understand that these peptides are going to be ranging from 2 to several dozens. So whenever we're talking about a peptide, that is specifically going to be smaller in size than a protein. Proteins are going to be much larger. Understand that when a peptide is written, the peptide bond is a single bond. So the resonance structure illustrated in the previous slide, pretty much it is just illustrating how dynamic that amide bond is in the context of a peptide. Now, these peptides are directional. What does this mean? That the peptides are going to be specifically written from the N terminus, which is the aminous terminal, to the C terminus, which is going to be the carboxy terminal. So here we have the example of a peptide. So in this peptide, understand that the first group that is present is going to be the amino group of the first amino acid. Okay, so that is the N terminus. And the C terminus, as you can see here, is going to be the carboxy terminus for the last amino acid. Okay, so whenever we have a written peptide, understand that there's always a pattern. And I know that this was explained earlier in the first video, so this is just a reintroduction of the system. So whenever we have a particular peptide, understand that the pattern is that we have a nitrogen bonded to an alpha carbon. That alpha carbon is bonded to a C double bond O. And then because we have peptides in between the amino acids, as you can see here, we have C double bond O N, alpha carbon, C double bond N, alpha carbon, C double bond N, alpha carbon, C double bond N, alpha carbon, and then the last C double bond O is the carboxy terminus of that last amino acid. So when we look at this peptide, we can see that based on the areas that I highlighted, this peptide is composed of 5 amino acids. So this is a pentapeptide because there's 5 amino acid residues. So looking at the yellow areas, you can see how the first one is tyrosine, The second one is glycine. The third one is glycine. The fourth one is phenylalanine. And the last one is leucine. So when it comes to a protein, understand that there's going to be many amino acids linked to form a polypeptide. The term polypeptide refers to proteins because they are going to be in a way a polymer of amino acids, and there's going to be many peptide bonds in it. So that's where it comes from. Okay, now understand, as I was mentioning, in terms of size, a peptide is going to be much smaller than a protein. And understand that in terms of molecular weight, a protein is going to weigh about 5,000 grams per mole or more. Understand that since they consist of one polypeptide or more polypeptides coming together, that's why they are so large. So to give you an example of a protein, here we have human insulin. In human insulin, it is a very interesting molecule because human insulin, it is actually made by two polypeptides coming close to one another. One of the things that I mentioned earlier in these slides is that the cysteine residues, so I'm just going to zoom in, for proteins actually form a disulfide bond. And they are illustrated, as you can see, right here in the molecule. So this is just showing the SH bond in the cysteine making a disulfide bond. But we will talk about disulfide bonds later in the semester. For now, understand that when we have drawings of peptides, drawing of proteins, when it comes to biochemistry, sometimes we have to use cartoon representations of the biomolecules that we're talking about just because they are large. If you take into consideration human insulin on the left side of the slide, you can see how it looks in terms of the functional groups that are present. And if we look at the molecule on the right, you can see that's the cartoon representation for it. It is much easier to see and comprehend when we look at the cartoon. So understand that both forms of illustrations are going to be utilized in this course. Now, taking into account what is happening in all the topics that we have discussed so far, now I want to review by doing a particular problem and to apply the knowledge of all the concepts that we have studied so far in this chapter. And for that, we are going to look at problem 21. As you can see, it says, consider the following peptides. And as you can see, we have peptide number 1, which has phenylalanine, glutamic acid, serine, and methionine. And we have peptide number 2, which has valine, tryptophan, cysteine, and leucine. Then, in the problem, it states that we are going to look at what are going to be the net charges of each of them at pH 1. We're going to look at the net charges of the peptides at pH 7, and then we're going to indicate the values for the net charges at those respective pHs. This is how we handle a problem like this. The first thing that you're going to use is table 3.2 from the textbook. You can obtain it from the slides or you can utilize it from the textbook. Now, understand that it is important that first we highlight the different amino acids that are present in our peptides. So that's the first thing I'm going to do. So using my highlighter tool, I'm going to select the information in the table that I have for phenylalanine, followed by glutamic acid, serine, and lastly, methionine. Then, when I'm looking at how is pH affecting my peptide, I'm going to look specifically at what are going to be the functional groups that are exposed in these pH conditions. Remember, when we have a peptide, understand, and I'm just going to redraw it, that the nitrogen in the first amino acid is exposed. And the carboxylic acid side of your peptide is exposed for the last amino acid. Remember that the bonds that I'm highlighting in blue correspond to the committed carboxy and amino groups for each of the amino acids, which is committed in a peptide bond. So once I understand that, then I have to take into consideration which one of my amino acids has a side chain that is affected by pH. In a peptide in which I have phenylalanine, glutamic acid, serine, or methionine, notice that the only one that has an exposed value is going to be, for a side chain, glutamic acid. So I know that my glutamic acid has a side chain that may be affected under pH conditions. Remember that a glutamic acid, the side chain is CH2. CH2, C double bond O. And then I'm going to determine what's going to happen with that side chain. So in the next step, I need to look at what are going to be my values for the functional groups that are exposed. So since the nitrogen in the phenylalanine is exposed, I'm going to look at the pKa value for my amino group on my phenylalanine. I'm going to look at the side chain for my glutamic acid, and I'm also going to look at the carboxy terminus of my methionine. Then I have to ask myself, at pH 1, what is going to happen to those functional groups? Keeping in mind that at pH 1, the general structure for my amino acid is going to be a protonated amino group and a protonated carboxylic acid. Then I have to tell myself, well, at pH 1, the pH is so low that my amino group in my phenylalanine, since the pKa for it is higher than the value of pH, is going to be an NH3+. For my glutamic acid, since the pH is lower than the value for its pKa, is going to remain as a full carboxylic acid. It's not going to be deprotonated. And then if I look at my methionine, because the pKa of the carboxylic acid is higher than the one of the pH that I'm looking at, then it's also going to remain protonated, meaning that it's going to stay as a carboxylic acid. So as you can see, overall in the molecule at pH 1, the overall charge of my peptide is going to be positive 1. Let's see what happens at pH 7. I'm going to be looking at the same numbers. So I'm going to redraw what is exposed. I have the nitrogen on the phenylalanine. I have my glutamic acid bonded to the serine, bonded to the methionine, and then the methionine has a C double bond O. I'm going to redraw the side chain of my glutamic acid, which is CH2, CH2, C double bond O. And now I'm going to ask myself the same question. Any value that is lower than the pH that I'm at is going to be deprotonated. In the first example, because all the values were above my pH, none of those hydrogens were going to be pulled. Okay? So at pH 7, I can see that specifically my side chain for my glutamic acid has a pKa lower than the pH. And what happens always is that if the pKa is lower than the pH of your solution, then the proton will be lost. So this oxygen is going to become negatively charged. The methionine carboxylic acid, because its pKa is lower than the pH of the solution, is also going to lose its proton. So it's negatively charged. The nitrogen in the phenylalanine, because the value of the pKa is higher than the pH, is going to retain its proton. So this is going to be NH3 positive. So when I look at the net charge of my peptide, understand that at pH 7, I have positive 1, minus 1, that is going to be 0, and then I have a negative 1. So the overall charge, or the net charge, for my peptide is going to be negative 1. Make sure that at home you follow those steps and you determine what are going to be the net charges for that second peptide at pH 1 and pH 7. The last topic that we're going to mention in chapter 3 is going to be section 3.5, which talks about small peptides that have physiological activity. Understand that hormones, specifically non-steroidal hormones, have actually an effect on the endocrine system of animals and humans. These small peptides are very useful, and as you can see, they regulate many cellular processes that include metabolism, growth, development, tissue function, sexual function, reproduction, sleep, mood. So understand that these peptides, even though sometimes we consider them just to be small, and in terms of size, you would be like, okay, what is their actual function? They are going to be, as you can see outlined in the first bullet point, very important. Now, understand that some of these important peptide hormones are cyclic, and they are cyclic due to the disulfide bonds that are going to happen between the cysteine molecules. And to give you examples of small peptides that have physiological activities, because sometimes we just think that, oh, only proteins have physiological activities. No, small peptides can also have some. The first one that we have is oxytocin. And as you can see, the structure of oxytocin is in the top. And that small peptide actually induces labor in pregnant women. Now, we also have another small peptide that is called vasopressin. And vasopressin, on the other hand, plays a role in the control of blood pressure by regulating smooth muscle contraction. When we look at the structures of these two small peptides, they're actually pretty similar in terms of the structure. So looking at all the different amino acid residues there, if we pinpoint what are going to be the differences between them, at amino acid number 3, as you can see, in oxytocin, we have an isoleucine, while in vasopressin, we have a phenylalanine. As we walk around the molecule, also on amino acid number 8, we have a difference between leucine and arginine, but pretty much, other than that, the molecules are very similar. So this concludes all the topics that we're going to be talking about in chapter 3.